Jandricic has over 20 years of experience in the integrated pest management of greenhouse pests. She has been the Greenhouse Floriculture IPM Specialist for OMAFRA since 2014. In this role, she helps guide and conduct the latest research on pest issues facing the industry, including both insects and diseases. And our second speaker is Judy Colley. Judy has worked with beneficial insects for over 30 years and with plant products as the IPM Technical Lead for 12 years now. Her work focuses on greenhouse floriculture industry, with tropicals being a highlight. She also consults with medicinal marijuana, greenhouse, and field strawberries. So I'll pass it over to you.
Great, thanks, Kara. And for those of you who don't know Kara, she's the greenhouse vegetable IPM specialist, so my counterpart and better half at OMAFRA. So I'm going to share my screen.
Thanks,
and make sure we do this right. Kara, how's that looking?
It might, oh, there we go. Looks good.
Okay. Okay. Yay. All right. So we're going to jump right into the workshop. So the reason we're doing this webinar is to sort of bring you all the information that Judy and I have learned from a bunch of different sources, and what we've learned from basically working with farmers on farm for almost 2 straight years now that have been struggling with this problem in Ontario, and try to put this information all in one place. So we've gotten information from the Netherlands, which have done some biocontrol trials. We're going to talk about some US stuff, mostly their great pesticide screening that they've been done that have really gotten us started on controlling this pest, as well as, like I mentioned, our on-farm trials in Ontario. And just to keep you apprised of what is going to be happening, we're going to be focusing more on more in-depth projects on biological control in both Ontario and Georgia. These projects are probably happening in the next year.
And everybody keeps asking us, why is the concern, or what all this hoopla is about? And just to give you some numbers, one customer of mine in 2 crops in the Anthurium and the Calathea has lost over 161,000 in wholesale value of this crop. And if you notice, we're going to talk about this later on, is they're just specific varieties. So this has affected, impacted one farm pretty substantially. And over the industry losses, there's been peppers 23% alone in crop losses. Peppers out in the field in Florida, 1.3 million. Mandevilla, offshore Mandevilla cuttings, there was crop losses in 2022 of 60%. 2023 was around 20, and in 2024, hopefully it's 10 and less. We're with the progress of what we've been making over the years. But the full losses in Canada and the US are still unknown because this pest is just being discovered in greenhouses now.
So we're going to talk about identification and monitoring because that's mainly the only thing we've got truly figured out from top to bottom. So I just want to open this by saying this is a regulatory issue, although this is unlikely to be a regulated, like a quarantine pest in Canada, because it's not thought to be able to overwinter here because it can't overwinter in the Netherlands. It's still a quarantine pest in certain counties in Florida, but overall it is considered a pest of concern by the USDA. So that means it is a threat in Canada to border trade. So if you have any parvispinus on your plants, you would obviously want to make sure that is completely cleaned up before you're shipping to the US, because there's always a risk that those shipments could be stopped if they're found, and they are sort of a noticeable thrips because they are dark and sort of bouncy. So these are the places where they've been found. So far, red is confirmed finds, and that data was shared to me by Cindy McKenzie of the USDA. And then we know for sure they've been found in Ontario, and then there's some other maybes of BC and Michigan. But the point is, it is being found in greenhouses throughout North America at this point. In terms of their appearance, you guys have probably seen this lots, but the females are a dark thrips. Their head and shoulders are sort of this like lighter brown, and then their abdomen is darker brown. The males look like sort of mini Western flower thrips. And they have these really bright red ocelli, which are the primitive eye spots between their regular eyes. They're almost like this like hot pink. And the only thrips that they really look like to me as more of an insect person is thripsitosis. But we really only find thripsitosis, that's pictured there on the right, on hydrangea. So most people are going to confuse it with these ones.
So just so you know, not every black thrip is going to be parvispinus. There's at least 11 that I've found in tropical plants over the years that I've been working. Just a quick reference, if the head is dark, it's not parvi. And if you're finding tons on a sticky card, it is not banded thrip or echinothrip. And just Sarah's put some nice 3 pictures for us to see the differences there. But just so you know, there's more than parvispinus that is dark. And coming into the winter, a lot of our ones that we're used to will turn dark as well.
Yeah, which is exactly why we added Thrips parvispinus to our Thrips identification key that's been made by myself at OMAFRA and the Vineland Research Innovation Center, specifically Ashley Summerfield and her amazing little graphic skills. If you don't find Thrips cute, you will as cartoon forms in this thing because they're adorable. But we've really made sure to add the dark thrips to this because after we discovered parvispinus in Ontario, we got inundated with vials of dark thrips because everyone was like, oh my God, is it parvi? And sometimes it would just be a dark morph of Western flower thrips or onion thrips. So this key has been tested. If you've got a microscope, you can do it. Yeah, I recommend everybody using this.
So there's really no difference in their life cycles between Western flower thrip and parvispinus, but the real difference happens when the temperature changes. So when we start to get our hot summers, 28, 30 degrees, our typical Western flower thrip and onion thrip will drop off, but then parvispinus will go through the roof and their life cycle is exponential. So this is where the most damage happens. And this is why it's serious for us to have a look at. So what we don't know about these thrips, really, all these tropical thrips basically, is their biology and behavior. So if you notice some of these tropical thrips, like greenhouse banded thrip, do the secretions do they contain any scent marking pheromones? And are they territorial and will they kill each other to protect it? Because I don't know if you've noticed, but whenever there's a population of greenhouse banded thrip, I can't find any other thrip there. And the greenhouse banded thrip is one of the dirtiest ones that I can find on a plant. So I'm wondering if they don't play nice, and because they do this scent dropping, that it deters other thrips from moving in. And then some other questions that we've come up with and we just ask ourselves is how fast can these tropical thrips adapt to other host plants? And can they complete their life cycle on these plants? Because from my experience, we had mandevilla in the greenhouse, we took them out, the thrips jumped to philodendron, but I could never find any larvae or other life stages on the philodendrons. And are they just sitting here until they die because they can't complete their life cycle? And then that leads to other questions like, in these scent droppings, can we make pheromone sprays, or can we develop more lures, knowing what these secretions contain, and actually will these lures act as a repellent instead of an attractant? Can we manipulate the parvispinus, force them out with an agitator and then spray? Are they eating mite eggs? And this was also thought to be happening as well. So a lot of questions still that we don't have testing done, and we don't have the knowledge to do. So
I, for one, am excited about the potential of poop lures or poop anti-poop anti-lures.
lures, yeah.
All right, are you gonna?
Is it frozen now?
Yeah, maybe. Are you gonna go with me? Here we go. Okay.
Okay.
Wouldn't be a webinar without at least one technical difficulty.
Yeah.
So the host list, I've made this, and there is a copy of this, a slightly older copy of this on Floriculture, my blog on floriculture.com, but this is still evolving. So if you look in the literature, the host list is very wide and very scary, like things like chrysanthemums and basically any ornamental we can grow. So far, that has proven not to be the case. But the original sort of like hot 8 plants we were seeing it on at first, Anthurium, Mandevillas, Gardenia, Hoyas, Schefflera, ficus, that has been slowly expanding as, growers have had this problem and it's bouncing onto other plants in their greenhouse. So some of these newer ones, something like prayer plant, there's a lot of damage happening there, but it seems to be only when it's planted right next to something parvispinus really likes. They probably won't necessarily eat it on their own. And there's also other ones, like we are seeing it on Gerber daisies and Garvinia now, but again, at really low levels, it's not really damaging the plant. It's more in the flowers. So the next step in this will really be to figure out which of these plants they're actually reproducing on versus, and, can continually continue their population on versus just, are they bouncing over there and damaging and eating it? So yeah, so we need to work on that. And they're also very variety specific. So we'll talk a little bit more about how you can use that to your advantage in the future. So when you're looking for this thrips, we've talked before about how damage is going to be your first sign whenever we do webinars or blog posts on things like this, but you have to remember the damage might look different by crop. So we've talked a lot about it looking like this heavy broad mite damage. This picture on the right on Schefflera, for example, that's sort of like the characteristic damage, but on plants like hibiscus and anthurium, it tends to look more like standard Western flower thrips feeding damage. So it's important to, if you're starting to see a lot of damage, either check the flowers, if they do have flowers, for dark thrips, or tap them and get them ID'd because it could be parvispinus on things like that.
So over my experience with tropicals, we've just collected some pictures to show you what the different damage does on all these different species. So Schefflera, the growing tip is really gnarled, and it goes all the way over to Ipomea, which we just found recently. But Ipomea grows so fast that it ends up with just crinkly leaves. And on Caltheas, as the leaf unfolds, you get dark margins on the one side of the leaf because that's the most tender as the plant is developing. And it opens up and it looks almost like low humidity damage, but except it's on one side of a leaf. So it's very unique kind of damage on these thrips based on how fast the plants grow as well. So now we're going to talk about monitoring.
So currently we are still recommending plant taps as the best method for monitoring. Oops, there goes my phone. So literally just taking your plants, like our example here with mums in the picture, and bashing them over a white pan. You can get really great ones at the dollar store, just like cheap cookie serving trays. Mm-hmm. Or Florida made this really great sort of cheat sheet to thrips about how you can recognize parvispinus. Parvispinus is the little itty bittiest one all the way to the right, but they have all the other dark thrips that you could potentially encounter. So you could put an acetate cover over this sheet and use this in the greenhouse as well. And that size would help you be like, oh, I should look at this because it might be parvispinus. It does help the first time you're doing it to have a hand lens, just so you can look at them more closely. We had a greenhouse where they also had a columbella infestation. And because columbella are also really small, dark, and pretty bouncy, for a second we were like, oh my God, there's so many parvispinus. And it turned out to be columbella. So, until you get used to it, because they are very different than Western flower thrips, use a hand lens. Look for movement because they are so fast. That's been a key for me, looking like even the larvae are just faster than any thrips I've ever dealt with before. And if you are going to use plant taps, to check the density in your greenhouse or which varieties it's affecting, or your control measures and how well they're working, you want to standardize your plant numbers week to week, because otherwise if we're doing different numbers of plants every week, it gets confusing. So that would be my recommendation there. And just tapping a plant, you're just bashing it over the white pan, trying not to break the flowers, although I'm not very good at that.
Okay, so threshold numbers, like a long time ago, we come up with, okay, 20 thrips on a sticky card, then we know there's a problem and you have to spray. If you have 20 parvispinus on a sticky card, your crop is probably already done. So Sarah's done a lot of trialing with what is going to be the new threshold for this thrip and what is an acceptable number with damage and action points at what threshold. So it's still a work in progress. Started off at 20, but it's certainly dropping fast. So if you see adults on the plant awash, is going to reveal some really scary numbers of larvae. Again, I have a very hard time seeing that larva. It is so small and it's very transparent. But that adult, as soon as it jumps all over the place. So you see something springy,
Yeah,
have a look.
I also have trouble seeing the larvae. I have graduated to reading glasses because of this pest.
Yeah. I have a 40 time lens, and I still take them home.
So just to give you an idea of how difficult even they are to monitor with plant taps, we're recommending that, but it's going to be based a lot on adult numbers, but then you're going to have to sort of like round that number up. And the reason for that is because when we were doing our plant taps, things didn't seem right at first. When I was first tapping the mandevilla, it seemed like the population was all adults. And it's like, okay, if this is all adults, how is this continuing to damage the plant after we do a pesticide application? And then we were also using alyssum as a potential banker plant or trap plant. And again, I was getting really excited. Oh, alyssum would be the perfect trap plant if it only attracts in adults, but they can't breed on alyssum. But plant washes are the gold standard in the lab, where you literally take a jar, fill it with your plant material, put alcohol or soap in there, shake the crap out of it, and then pour it through a series of sieves. And then you're just left with the thrips on a piece of filter paper. And that shows you what's definitely in there. And it was kind of frightening that we were missing 30 to 65% of the population as larvae when we were doing taps. So, if you're tapping out, 6 adults on a plant, you can probably assume that your pressure is actually 10 or 11 thrips per plant if you are considering the larvae you're not getting. They seem to be just very sticky. Like you can see them running on the growing tips, and then you'll hit the plant and they'll be like, I'm still here, I'm fine. And I think like most thrips, they also like to bury themselves in the little growing points. So sometimes it's just not able to tap them out, especially in a trellising plant like mandevilla. So just keep that in mind when you are setting thresholds for your own facility. People want to know if cards work. They work very well for this species. Like I said, they're bouncy. Every single one of these red dots is a Thrips parvispinus, my student I had to count out last year. I would say they're not useful for initial detection because, like Judy said, if you've put cards out and you find 20 parvispinus on a card, that's like finding flying aphids on a card. Like it's already too late. So what you want to look for initial detection is really damage, inspecting flowers because they seem to love pollen and nectar. If you're worried, even do plant taps and washes. Judy was just telling me some cuttings coming in from places. She's been doing plant washes and taking them home that way to sort of figure out the baseline of what's coming into a greenhouse. You can use trap plants like as an indicator plant. We'll talk about that a little bit later. But cards can be used to monitor infestations after it's already shown up in your greenhouse to tell you where in the greenhouse and potentially some information about density. But there's a question mark next to that because we don't really know how it super relates right now. This is data I got from a working greenhouse where over the weeks we did plant taps, which is the orange line, and the blue line are the cards we had in the bays, just put out sort of at regular densities of monitoring cards. And what we learned from this is like the card numbers do somewhat correlate with the tap numbers, but cards were definitely slower to show decreases. So the first year we were doing this, we set the economic threshold at 10 thrips per 5 plants. These were 6-inch mandevilla plants. And so when we hit that threshold at week 48, we sprayed, and you can see that after we sprayed, the plant taps showed that the numbers on the plants went down really well. But if you look at the blue the blue line on the cards, it looks like they exploded. So if we had just looked at cards, we would have concluded that our sprays didn't work and we probably would have kept spraying. On the flip side, the cards actually seem to detect the thrips coming back after the pesticide residues had worn off. The cards seemed to notice that the thrips were coming back earlier than the plant taps did. So overall, we recommend definitely doing plant taps after pesticide applications to confirm kill, to make sure what's actually on your plant, and then keep an eye on both post pesticide spray when you think they might be coming out of that protection sort of time.
And we're going to talk about this after a while, because if you had gone in with just the sticky cards at blue and sprayed again, then it's going to build up more resistance. So we'll talk about this a little later on as well.
Yeah, absolutely. So this is something I'm working on right now, a path to easier monitoring, hopefully plant tapping, especially when you've got all the varieties in the greenhouse and they like different varieties better than some, and some varieties can handle more damage than others. So there's difference in attractiveness and difference in susceptibility. So you have to tap a lot. So we're trying to find an easier way to make sure growers can get the information they need. So we're looking at things like maybe cards plus plant inspection. So maybe something like how we monitor for Bermesia whitefly and poinsettia. We don't sit there and count every nymph. We pick up plants and we say infested, not infested. And then you work from a percent plants infested in your greenhouse. And you know that if it's over a certain level, you need to intervene with pesticides. I would say that that maybe would work better if your infestation is low or on non-trellising plants, because I think on the trellising plants like Mandevilla, if any of the thrips are hidden in those little growing points, you picking up a plant isn't going to show you much. You would still need to inspect things like growing tips and flowers and foliage more closely. So that's one idea. I think I'm more leaning towards this right now. Cards plus damage count. So with the Mandevilla, what we've been doing is we've been putting cards in pots, also tapping those plants, counting the thrips on the cards, and then checking the growing tips for damage and just scoring them as sort of 1 to 5, like 1 being destroyed and 5 being like you're growing and you're trellising properly. And that seems to be telling us more about whether we should be intervening with pesticides or not. It's more work than, just plant inspection, like the presence absence. I think it's less work than plant tapping though, and probably more accurate in terms of damage. So we'll just have to see how that goes on. But those are my suggestions so far. Test them in your own greenhouse. Please give me feedback on how it's working and if your staff can do this on a logistical way, because it is, and financial way, because these guys are hard to monitor for. Okay, so the next thing we're going to talk about are mechanical and cultural controls, and Judy and I need to make matching t-shirts that say traps are your friends, because we both really like sticky cards at this point.
So I get a lot of people saying I don't want to put sticky cards in my greenhouses because they're unsightly, and when I have tours come in, it shows that we have bugs in the greenhouse. But you have to remember it's one of the cheapest ways in your greenhouse to control thrips. You're not using a pesticide, but it's taking place of almost using a pesticide. It'll reduce the thrips population for you from 17 to 30% without you having to do anything other than put them up. And there's many ways that you can hang them so that they're not unsightly. And you can see them in the pictures. Some of my customers are very creative with how they hang them. And it's necessary for a complete IPM program, especially with these thrips. We know they go to the sticky cards. So the more the merrier too in your problem varieties. If you want to get them to go to the sticky card instead of in your plant, again, you don't have to count those every week. It's just put them in and let them trap as much as possible.
Yeah, and they seem to kind of go in waves. So, those pictures where they're, a single card per pot in a problem variety, that's helping prevent it from spreading through the greenhouse. And also those numbers Judy mentioned of 17 to 30%, that's what we've actually calculated from our on-farm trials. So those numbers aren't just coming out of nowhere. So for example, this was the first year I worked with this grower with Mandevilla. We were comparing a couple of cultural controls or mechanical controls, I should say, pruning with mass trapping and also biocontrol to just pruning and biocontrol on the white variety. And what we really learned from that is that the ones that had the mass trapping in them, we had much better control. We had over 25% more control in those than the other variety. So we knew we were onto something and we carried that through. And we've done a couple of tests. So I worked with a couple of different companies to test different colors for this facility, I have to say, because we do know from doing other tests with mass trapping that the thrips response to color really varies by greenhouse location, cover type, light quality, because it really all has to do with how they perceive the color, and that's affected by light and hue and all that fun stuff. So what we found in this one location was that yellow worked better in the summer, but this really dark blue one worked better in the winter. And then of course, Judy's found completely different results in her other facilities. So again, basically just get your hands on all the colors you can. We were really excited about this green that apparently in the Netherlands works really well. didn't seem to work for us at all, but
Works great on fungus gnats.
yeah, yes, we did get a lot of flies for sure. So, you know, if you can get your hands on a bunch of colors, it's worth testing.
So another way for mechanical control is physical barriers. And just from a fluke back in 2018, a customer of mine was having trouble with Hoyas. We didn't know at the time that it was parvispinus, but we could see the thrips on there. And I thought, okay, let's use some strawberry cloth to see if we can protect these to keep the thrips off. And then we threw aureus underneath the cloth. And it was successful at continuing this Hoya crop. So now my Hoya grower is using this strawberry cloth or landscape cloth on all the Hoya cuttings, and aureus is being put underneath because we know that parvy's coming in on them. And if we can trap them and make a little mini environment where the aureus stays in there and keeps other thrips from bouncing into this crop, we can grow a nice Hoya from start to finish. The Netherlands has also been experiencing with hanging some of this cloth as curtains from the floor up to the knee height. And as you saw on the one sticky card picture, that clothesline, you can just hang it like a clothesline in between. So another way that we can do this, it'll also prevent movement from your more hardy varieties that don't show all the damage versus ones that do. So if you can keep the movement, keep these thrips from bouncing back and forth. Like for instance, if you had the Calthea in one range and you had the Anthurium right beside it, you could run this cloth down and it would stop the parvy from jumping over. We know they're lousy flyers, but we know they do like to jump. So again, it's another way, it's cheap. And if you can keep your crop healthy, then by certainly adding this physical barrier in there would help.
Yep. Every time we click the screen for something
Yeah.
else. So I think I just need to, oh, there we go. Okay.
So we've got this dilemma or debate, I should say too, about trap plants and banker. For me, banker plants for your biologicals or your beneficials inside the greenhouse do not work. The difference is between indoor and outdoor light. We have seed versus the proven winners, which is a vegetative cutting. It doesn't produce pollen, but it produces nectar. But inside, they do not produce the nectar quality that beneficials really like. You take those same plants and you put them outside, and it is unbelievable what you can trap as far as native beneficials that come in. So to try and put alyssum in a greenhouse to put aureus on and use it as a banker, you can put 100 aureus on one banker plant and you're lucky to find one. And you're lucky to find multiple generations. So we flipped that around to opposite thinking based on trials that I have done and that Sarah has done in greenhouses. We're now using them as trap plants. So that led to the discussion, can we use alyssum or susceptible varieties or cultivars to trap the parvy and then throw them away? Very similar to what we used to do with mums a long time ago. So that's something to think about. We do not want to use them as a banker plant for your beneficials. And Sarah has collected data.
Yes. So I went into this, you know, we knew from information from Belgium and the Netherlands that the big bios were really going to contribute most to biological control with this pest. More on that later. So we knew we wanted to get aureus going, but aureus is obviously expensive in floriculture crops, especially in Canada. So how do we boost those numbers? So we thought about using banker plants. So we did Garvinia, which is like a long lasting plant, as well as alyssum in the same pot. And then we monitored those. And just as Judy said, I probably should have called Judy before I even started this. The return on investment of our aureus was nothing. We were putting 50 aureus per plant, like one of those big pots. We did that twice and we got maybe like 2 or 3 aureus per pot back. So that didn't work out super well, although we could find them in the crop. Yeah. So they were happy in the mandevilla. However, what we did find was a lot of parvispinus. So on this graph, this is the experiment, the week of the experiment. The blue line is the mandevilla. So again, our crop economic threshold was around 15 thrips per 14-inch pot. Again, probably too high, but that's what we were working with at the beginning. The orange, the lighter orange line is the alyssum, and the darker orange line was the Garvinia. So you can see that the Garvinia really wasn't that much more attractive than the mandevilla at first, but even at the second week, the alyssum was twice as attractive as the crop plant, which is what you want out of a trap plant. And then by week 3, it had exploded. So think back to that wash data. That's exactly why we washed them, because I was like, they can't be this attractive. This, by week 3, you could actually be getting reproduction on the plant. So we started washing them, I think, in week 4, and that's when we realized, oh, they're also breeding on this. So if we are going to use them as a trap plant, we do need to throw them out or use pesticides. So that's what we did in this green box. So by week 6, when we realized there was bonkers numbers of thrips on the alyssum, especially, we sprayed them with success, and then that dropped the population down about by 50%. And then we just removed them. And by doing both of those things, it was the first time the thrips on the mandevilla dropped to almost 0, because we were just taking so many thrips out of the system, and they were well below economic threshold, and they stayed that way for a little bit of time. So unfortunately, we weren't able to replace the plants and start over, just because the plants had to move to a new location. But it was a really good proof of concept that this can work.
And you found that they were attractive even without flowers on them.
Yeah. So we also did this like in the winter when they weren't flowering, and they're still more attractive, I think 2 or 3 times more attractive than the mandevilla without flowers. We were also pulling the flowers off the mandevilla to reduce their attractiveness as well. So we would need to do this like with a full flowering mandevilla crop and a full flowering alyssum, just to be sure. But that's another good way to make your trap plant more attractive, is make it the most attractive thing in the greenhouse.
So thinking outside the box, if you're using your alyssum as trap plants, seed alyssum is very quick. You can sow it, it comes up in the pot. You don't have to transplant it. Again, it's attractive without the flowers on it. So you can have a rotation of this seed alyssum going. On the left-hand side is a proven winners. These are vegetative, so you pay some money for these. So maybe you could use these as your test plants or your spray plants, or ones that you're going to spray and clip and then wash your clippings to see what you're actually getting out of there, instead of replacing them every 2 weeks. The key is to replace them or spray them so that you don't have parvi breeding on these plants. And then the blue sticky cards, it'll give you the presence or absence of thrips. And you'll be able to tell with your sprays, if you're spot spraying them, your percentage of kill as well. So yeah, instead of replacing them every 2 weeks, if you've got proven winners, you can use them and cut them back. But again, you have to have rotations of ages of plants then in your greenhouse.
Yeah, so you could really use them one of 2 ways, as an indicator plant, like Judy said, and then you would only need like 1 big one per variety or something, or maybe 3 down that whole row. But if you're going to try to use them as like to suck thrips out of the crop, then you need a lot bigger density. So we were doing 1 full 14-inch pot of alyssum per every 50 crop plants. So it was quite a few. So eventually those big pot sizes got too big, so we did like 4 7-inch pots instead. But that's the kind of density you need to evenly suck them from your crop if you've got a problem going.
Yeah. So another thing that we found out, going way back to the beginning slide when we told you about crop losses. So on the left-hand side are these plants are grown in the exact same greenhouse, basically 2 feet apart from each other. So to me, variety selection is key, because if you can grow these, yes, they will have parvispinus, but if they don't show any damage, then you're going to have a crop that you can sell. And this is one that we can maintain a lot easier versus one on the right that you're going to have to spray all the time. It might not finish at all. You might have to throw it away, and it's very susceptible. Or can we use one of these susceptible varieties as a trap plant and put it in the crop? So there's some things to think about, but if you're going to do that as a trap, remember you're going to throw these out. So get like 100 plants in and then throw them out. But variety selection is really key here.
Yeah, and we see that in mandevilla as well.
Yeah.
So biocontrols, not your normal Western flower thrips biocontrol program, which we've basically learned for every thrips that isn't Western flower thrips, even onion thrips.
So we've gone at this is that we're rethinking the bios. We are not veggie bio programs. This is not working for us. So we have to rethink this. We know that they are not going to establish, except your ground ones like Atheta and Hypoaspis, but you got to treat everything else as a weekly introduction, especially aureus. Mechanical controls are going to be one of the valuable assets to this program. And people say, well, I get too many bios stuck on my cards, but that's not true. Again, Vineland has done a lot of research on all the different colors of sticky cards. And you can see the highest, it looks bad because it's a long graph, but the scale is like less than 2 per card per week of Aureus. And it was the blue that was more attractive. So if you can trap Parvi on a yellow card, you know, switch colors. Again, the colors are going to depend on your greenhouse. And you can see that you're really not going to impact your bio program with putting sticky cards everywhere. So then I also get the questions, but Aureus diapause, so I can't use Aureus in my bio program. But I just want to reassure you that Aureus reared commercially will not diapause. The key is the 4th and 5th N-star stage, and that has been bred out of them during the rearing process in our labs. The key to know is that when you're doing weekly releases of Aureus, it's going to be their offspring that diapause. But again, I'm not looking at reproduction in the greenhouse of Aureus. If it does, it's a bonus. The biggest thing is to get Aureus in there because it is going to eat a lot of thrips. And again, it's the best one for all of these tropical thrips. And so how do we get Aureus to last longer? Well, BioBest has done all these different trials of food and which one works the best. This is one of their many slides, and it just proves that feeding them Hephaestia will get them to do almost 800 thrips in their life cycle versus without just thrips by itself. So it's kind of like the spinach for Popeye. This Hephaestia is for Aureus. So it supercharges them and makes them live longer. And if they live longer, then they eat more for you. So we also taken all of the bio programs that we've gotten from all over the world. We put them on a chart for you here. And some of these numbers are scary high, and it's just not feasible for Ontario biological programs to be using them at these rates and at these high of numbers. Because even when I first started doing my program, I was using Aureus at 5 to 10 per meter squared, or 0.5 to 1 per square foot. And that still was too costly. So we're always looking for ways of bringing this down. But just to give you some numbers, they are pretty scary, 40 eggs per plant. We just can't afford that here. And some of the big differences now is Anthurium crops versus Mandevilla. We know they're both coming in with parvispinus, but what makes them so different is the characteristics. Anthuriums are a shorter crop. They are grown at 20 to 25, which is optimal for your bios. And the growth pattern of these plants are a vase shape. So anything that you're going to spray from the top is going to go down and run down in the plant and sit in the leaf axils. The thrips have to come out sometime, and they're going to come into contact. So this crop is a lot easier. And again, their pollen is not, they don't produce pollen until they're well past the cell point, and the pollen is very big. Whereas Mandevilla, it's a completely different plant. It does have a latex sap, which maybe affects the bios. We don't know. You're growing at plants that are longer cycle. They grow different temperatures. And then the hardest part for us is growing at temperatures quite cool in the winter time at low light levels. And they trellis. So are we putting enough bios in when they're small? But then as they grow, do we need to put more bios in? And then again, the cost will increase. And the difference we're going to talk about today is Mandevilla cuttings that are sourced from Ontario or outsourced. And we're going to come up with some big differences for you. So in 2022, 2021-22, I started Anthurium biocontrol trials for thrips in the Anthurium. And this was before I had a problem with Harvey. My biggest problem was echinothrips in these, and then it moved into Schultzy. So I had a couple of different thrips in there, put Aureus in, cleaned them up, but then all of a sudden this new damage started to happen. Couldn't figure out what's going on, cut a plant back, did a wash, brought it into Ashley, and Ashley from the Vineland, she said, oh, this is Parvi. So then now what do I do? Because I know that at this time, the predatory mites were not effective. I started with cucumerus, but I know that the brand mites can establish in Anthurium, so I didn't want to use that too long. So I switched to Zwirsky. They all got hypoapsis at planting, and then we were doing nematodes. Didn't see any improvement although Echinothrip and all the other thrips weren't there. It was just parvi that was left. So that led me to think, okay, how can I do this? I know Aureus works on tropical thrips. Will it work on parvi? So I took all of that money from nematodes and Zaworski and put it into high levels of Aureus. And then I didn't have a rate to go by. So I thought, okay, let's do the bio program rates for vegetables. And we put in 1 or 10 per meter squared for 6 weeks into pepper plants. And then we drop it down to 0.5 or 5 per meter squared. And again, weekly releases were continuing all year round. So I was doing mass trapping as well with all these different colored sticky traps to see what I could catch. And I figured that the sticky cards were related to almost 30% reduction in adults. And then soft oil was being used at shipping to knock off any adults and then shine the leaves up on the anthuriums. So the outcomes of that, it was successful, but it was very expensive. And I still had those unsellable varieties that we had to throw away. And that's what led to that number of 161,000 in losses. So in 2023 and the beginning of this year, my successes with the anthurium crop, that I know that they're all coming in with parvispinus, and some varieties show damage and some don't. By doing these sticky card trials, I proved that there was just as many in a red variety that didn't show any damage versus the ones were coming in, we knew. All the new plantings in the first 5 weeks are treated 7 days apart for parvispinus with Lalgard M52 and 2 applications of success. And both were used at 1 milliliter per liter rates. And so far, even in the purple that we never got rid of, we got rid of the Ariza varieties. They're coming out nice and clean. So after that first initial spray, we go in with Aureus, and it's the heavy lifter. I was doing it 1 per square foot until we started with the Lalgard, and I've been able to drop the Aureus to 0.25 per square foot, as long as we keep continuing with the Lalgard and success. So again, we still finish the crop 1 week before shipping with soft oil, just to make sure there's nothing on the leaves and to get rid of the spray residues if there is any. And to try and bring down the cost a little farther, I'd like to try Botanigard EC at the end or some point during this crop to see if it would work. So now I'm going to talk about Ontario mandevilla cuttings. And please know we've got 2 ends of the scale here with this talk today. I'm talking about the Ontario sourced cuttings, which are the easy ones. Sarah's going to talk about the bad ones.
I know, I'm a little jealous through this whole talk where she's like winning in Anthurium, winning in Ontario mandevillas.
Okay, but yes. So since 2016, I've been doing a mandevilla crop. The bio program has evolved as pests change and come and go. And I find because these plants keep growing taller, and I know that Zaworski and cucumbers don't work very well on this, I switched to flyer bio program. So I used all the flyers that we have out there. And the cuttings come in clean, which is what I keep enforcing here. But at the facility and other ranges, there is parvispinus. Anthurium is being grown there. So I know that it's going to come. So we do a preventative program. So all plants received 1 application of Lalgard M52 at planting. And that rate was 1.1 milliliter per liter. I'd like to try it at 0.5 to see if we get the same control again to help the grower reduce the cost. So far, the aphids have been the biggest problem, and the crops have had 2 bee-leaf and 1 ventigra. I check the flowers and tap them out. There is a biological company as well, or a scouting company that comes in and also does the same thing as me. So we're on the same page, but we check the flowers and do plant taps. I will also go in and take cuttings of the ones that looked deformed and do a wash when I get home. In the varieties that we are suspecting that we have parvispinus, we put a proven winner, Alyssum in there with the blue sticky card. We're using it as a trap, not a banker plant. And then I'm looking for presence or absence of parvi, and we change them every 2 weeks. So far, I've been able to reduce the Aureus to 0.2 per square foot or 2 per square meter. And the cucumber sachets are put in baskets primarily for broad mite control. When they hang up, we can't physically see up there. So it's sort of a peace of mind for broad mite control and other thrips control that fly in through the vents. So if we find parvi where we did, and I suspect that they were coming in on the workers because they were sort of just in the first rows, a foliar application of Laugard Men 52 was applied. So far, only 2 parvi were found in the flowers in early December. Met 52, Laugard 52 was sprayed. And where they were sprayed, there's been no parvi in the flowers since the second application of M52. A dark blue sticky tape has also been put in the varieties where we found, again, to stop positively thinking that they're going to jump from one variety to another. There has been no visible damage on the mandevilla at the beginning of December, but there was parvi on the sticky cards in the trap plants. So I was able to keep the aureus at 0.2 per square foot. And at the end of January, in the last group of washes that I've done, I took samples off of suspicious plant growth, and I didn't wash out any thrips. So again, that confirms maybe that the damage is old. And now that the plants are cooler, they're taking longer to grow out of this damage. But that the MET 52 application probably did help. So I will still continue to do washes weekly. Okay, now the nasties.
I just want to go back to this one picture.
Oh.
Judy
Oh,
sent me
yes.
this picture and she was so excited. She was like, look at the aureus and the tips eating the parvispinus. So yeah, they do get in there. So that is exciting.
Yeah.
We have to focus on the happy things. So the sad part of the story, offshore mandevilla. So these are ones coming in from Guatemala. They're coming in infested. This is the 3rd year in a row, and every year one variety seems to be more of a problem, but really all the varieties are coming in somewhat infested. However, we were doing dips of all the varieties when they were coming in this past summer to see if we could figure out, which varieties were the heaviest and the heaviest infested. And the interesting thing we figured out, and I should credit Vineland Research and Innovation for this, Ashley helped with this, was that we found very few adults and larvae in the dip water, unlike we do for Western flower thrips when like mum cuttings come in. So we suspect they're coming in as eggs, which makes it hard to do plant inspections and make sort of any conclusions off the cuttings you're getting in. And these are the growing temperatures, like Judy said, reasonable temperatures in the summer, but grown quite cool over the winter, which can interfere with biocontrol a bit. So our project goals for this year were to really try to give biocontrol a chance. The previous year, we sort of mucked around a bit with biocontrol, but we ended up doing a spray program, which was successful. And that's how we dropped from the 60% losses in the first year to only 20% losses in the next year. But if we don't have to rely on purely pesticides, we don't want to. So the grower, I really have to give him, and the owner a bunch of kudos for being willing to try this. So we wanted to use really high rates of bios if we could, minimal pesticide use except for spot sprays. We would try to base this on threshold, but also try to avoid pesticides as long as possible with a drop dead date of December 1st if things just weren't working. And that was based on our experience last year that if the plants weren't really putting on growth by December 1st, they were not going to be ready in time for sale in the spring. And then we also wanted to compare 2 types of biocontrol programs, which I'll show you. And again, using sort of a Bemisia program as our model of trying not to do pesticides at the beginning of the crop, knowing we could possibly wait out resistance and the pesticides would work for us when we needed them to in December and onwards. So our initial plan, like any good Thrips biocontrol program, should be to start in propagation, right? Like you want to put on, front load your bios, low pesticide residues, all that good stuff. So our plan was to do Swirsky, Botanigard, and nematodes, all of these ones that preferred higher temperatures. So Carpocapsis is better at higher temperatures than Feltii, and according to some studies. The problem was that the mandevilla in propagation were tented. And we knew that basically once the temperatures went consistently over 35 degrees Celsius, none of these bios were going to work. So we put temperature and data loggers inside the tents. And basically after the first month, there was no point in adding anything. So we stopped doing biocontrol. In retrospect, maybe we should have replaced that with a soft spray of something, but we did not. And then after we took the tents off, 3 weeks after we took the tents off, the numbers just exploded. And that's about the life cycle of, when we do Western flower thrips experiments in research greenhouses, you put the thrips on and you keep waiting and you're like, oh, nothing's happening. Maybe we didn't put enough on. And then week 3, it's just like, kapow. So it sort of tells you they are there as the egg form. There's a few hatching out, and then just as soon as the temperature is right and their populations build up enough, they just explode. So what we did do is we dipped those rooting cuttings in an oil dip, so Suffoil X, before potting, and that did reduce the thrips population by 70%. So in terms of the 2 biocontrol programs, the first one was our high bios and trap plants program. So this is just our like go crazy program in terms of like, let's not worry about expense. Let's not worry about labor. Let's just do it all and see if we did everything. Could we have a curative biocontrol program, knowing that these are coming in dirty, knowing we couldn't do that much in prop to lower it. Although we did start at fairly low levels at potting at week 37, we were under 5 thrips per pot in these 6-inch pots, which was much lower than our original threshold of 10 thrips per pot, you know, when we first started looking at this. And other than this one little blip on week 39, everything was well under threshold. So we were like kind of excited about it. Our average is less than 4 thrips per pot. Getting into fall, it was less than 2 thrips per pot and tap outs. But as you can see, the rates were really high. Aureus, we were doing 4 per meter squared weekly. We had a theta in there, we had hypoaspis in there, and we had tons of trap plants, 1 big trap plant per 50 crop plants replaced every 2 weeks. And you can see by the little green dashed line. So all the other lines are different Mandevilla varieties, and you can see that some are lower than others. And then the dashed line was our alyssum, and you can see that it was always higher than the crop plant. So it seemed promising, but we'll get there. Program 2 was our more economical program. So low bios. So we had some Aureus in there, but it was only 2 per meter squared. We threw some Atheta and hypoaspis in there weekly. And what we were hoping would be the heavy lifter would be Bioceres EC weekly at a high rate. And things were trucking along pretty good. These were bigger plants, 12-inch pots, which is why the threshold was set to 10 per pot, although that also was probably too high. Things were trucking along nicely until week 36, and then things started to explode, especially in a certain white variety that has been the bane of my existence.
And that's August, September, which
is
hot.
Yes, right? Exactly. Once it was hot.
Mm-hmm.
And we had started near zero at some point because we did our last bit of DDDP on week 32. So things seemed like they were good. Mm-hmm. And then the heat came and it all went out the window. So the one positive thing about this was after this outbreak, we used MET 52 sprenches at the high rate and also added anistis. And probably it was the MET 52 doing the heavy lifting, but we were able to bring this outbreak down to basically almost zero in just a couple of weeks. So that's amazing for a biological pesticide. We did near the end of this, week 43, just fully clean up the whites with Belief and Rinon. So softer chemicals where we could still keep going with our biocontrol. And then again, things were looking really, really good towards the end of the fall. But growth was just not there. So if you saw the earlier pictures that Judy had from the one greenhouse that's working on a more preventative program, you saw they already had some of the trellises in there, the plants were growing. These had, you know, the yellow circles, they were trying to trellis real hard. And then you've got these other red circles where they're just like nubbins and they're being eaten. And so when we compare the damage at both programs had to switch to sprays in late November, basically the same as happened the previous year due to lack of growth. Overall, the growth was a bit better in the bios plus microbials program, but they were grown at different locations. So maybe production has a little bit to do with that. And what we did learn from this is the actual damage threshold is obviously much lower than 5 thrips per plant, since neither program could keep up, especially once the temperatures got cooler. We had fewer thrips, but somehow more damage. So when your plants aren't actively growing and can't actively grow out of that thrips damage, and there's only maybe like one growing point happening at a time, and that's getting chomped, and then the plant can't grow. So I think we need to change our action thresholds depending on the time of year as well. In terms of cost, obviously the bios only program was more expensive, both in terms of labor and product, but neither program was really economically feasible yet, which is why I'm not going to get into the actual monies. But overall, thrips pressure between the two programs was similar, although the high bios program with the trap plants was more stable. So we learned a lot from this, things we can play around for next year. some of the lessons we have learned are you can never have a period of nothing, even when you feel pretty good about yourself. So again, after that DDVP application, these are 14-inch plants that we were also trying biocontrol on. So week 32, we did DDVP, cleaned them right up, started talks with biocontrol companies about donating products for experiments. You know, by the time the product comes in and we decide what we want, it's been 3 weeks and things went bananas. Things went off the rails. And I have to take full credit for this. It was like, you know, obviously this is a team effort between the grower, the owner, the biocontrol companies. We're all trying to figure out what to do because nobody's really ever done this. But, you know, I was the captain of the ship here and I should have never allowed this to happen. So obviously at this point, we went in with pesticides. We were able to clean this crop up. And it's been pretty stable with pesticides since. And I think this crop is going to come out fine. So thank goodness. And another lesson we've learned is that biocontrol just may not be possible in the winter. I might even need to set that do or die date of December 1st back to mid-October. That's kind of what we do with poinsettia and Bemisia. You know, if by the first week of October, you don't have your Bemisia under control with biocontrol, you got to switch to pesticides to get that under control before sale. It might sort of be a similar thing here. And part of that reason is just we already know that low light, low temperatures, the quality of the light affects bios. So this is a graph taken from Stephen Jandricic, who actually, I believe, discovered Beauveria bassiana, where if you sort of on the x-axis look at temperature and compare that to percent optimal growth rate of different kinds of fungi, so Beauveria met 52, other things, they both sort of, all of them sort of tap out at just under 20 degrees, so around 18 Celsius. You only have 50% growth of your fungal pathogens, and that's not going to get you anything. So there's not much point, like, you know, he's the guru of this all, and he says, yeah, below 20, there's not much point. And above 30. Yeah, and above 30. Good point. Thank you. Yeah. So, and as I mentioned before, also at that time, the plants are growing slower and they can't outpace the damage. We do know, however, that parvi keep going anywhere above 7 degrees Celsius, and that's information from the Netherlands. So, like I said, pesticides may always be needed from October to late February if parvispinus is already present. But we do know that some bios and microbials, if you use them early in the production cycle, will preserve chemicals for you when you need them. Because so far if we've done bios first and let it go for as long as possible, all our chemicals have worked. So that's good. Lastly, and I won't harp on this too much because I mentioned already, our thresholds still need work, especially in the fall and winter. I think this has to be a lot lower. Maybe if things are looking good after pesticide sprays, you can monitor just the Thripsiest variety and use that as a metric. Thripsiest meaning the ones that they seem to be most attracted to and will reappear first on. I think that could be useful. And just a reminder that larvae don't tap out well, but they do seem to be responsible for the bulk of the damage. So a better way to monitor for larvae would be great if anybody figures that out.
So chemistry, this is always fun. So the first thing is, we always stress this for every crop, for every pest, is to dip your cuttings. And there's been plenty of research. Rose has done a very extensive dipping trials with lots of different combinations. Most of my customers use Botanigard and Sufoil X mixed together. But, you know, your percentage of Sufoil in a dip is going to be a lot lower when you're dipping a very soft cutting than it will be later on. So you want to expect your cuttings, but like you said, we can't really find parvispinus on the cuttings. So we're assuming they're coming in as eggs. So you want to dip them all and then set around 17 days later after they have substantial roots or you're doing a first transplant, you want to do it again. And this time you want to make sure it's going to be in an oil and it will also kill the eggs. So dipping is a very important step. Biorationals. Don't forget about these. There's no known resistance to these at all, and they include them in your chemical rotations to break up the chemistry and the chemical groups. They'll add to your list of AIs, and then you want to make sure you're spraying at your optimal temperatures for the best effective use of these.
Yeah, so this is some of the work that Dr. Alexandra Ravinthi from the University of Florida. Yeah, we love her. She's our new favorite person because she's done all the preliminary screening of both the pesticides and some of the biorationals in here. So she was the one that let us know that Bioceres EC was of the microbials one that was working really, really well. Other EC formulations haven't been tested yet, like Botanigard EC, so we do need to follow up with that. But you can see the difference between Bioceres EC and Botanigard WP are really different. So like larval percent death, both direct and indirect mortality is around 50 to 100% with Bioceres, but it's only 20 to 40% with Botanigard. So, you know, again, back to this isn't your dad's Western flower thrips program. You know what I mean? We're so used to using Botanigard WP for everything. And in this case, it's not going to work for you. So think about EC formulas. Stuff oil X, like Judy said, is working for some people in Mandevilla. We're not seeing that as effective except as a dip. And then one thing Judy and I have noticed worked really well is the Lalgard M52 as a sprench. It hasn't been formally tested yet, but it seems to be working very well on farm. So with any of these, you know, preliminary screenings, like they're done in a lab. So the next step is obviously to move up to whole plant trials. And that's some of what we're bringing to you with these on-farm trials, but we need to do that more formally. And I know there's a project happening between the IR4 program and Lance Osborne and Cindy McKenzie in Florida. And we're doing sort of a project at the exact same time with the Pest Management Center, or PMC, with Flowers Canada and Rose Boughtonhouse at Vineland, and I'm advising a bit on that. So we will have actual, how well do these work on plants and different kinds of plants, aka Mandevilla, because they're annoying, and another plant at least, and that will be useful information as well.
And just to top, like the Bioceres EC or Botanigard EC, if they look at the rates of control, 80% kill in your adults. So instead of using success, use that, you know, so then you're not going to get resistance from this, but you will with success. And if in Canada, all we have is success and a pylon that really are effective, we're going to be in trouble. So just, you know, don't forget about these. So the first line of defense, when you're starting to get these cuttings in, like whether it's Mandevilla or whether it's Anthurium or any of these other tropical plants, you want to use, they don't have very good roots on them yet. They're very delicate. You want to use your biorationals here. So like your Lalgard M52, Botanigard WP, Bioceres WP, Safer Soap, or Suff Oil. And you can mix the WPs with Suff Oil, but again, watch that rate. Make sure it's very low, almost the dip rate, so you don't get any phyto issues. The second line of defense is once these cuttings have a decent root system on, whether they put right into the finishing pot or whether you're going to put them into a 4-inch before you put them up, that's when you can switch to your oils. So again, Lalgard M52, Botanigard EC, Bioceres EC, and Suff Oil X. And again, these oils are going to be better for you at controlling the eggs.
And these are the chemical products, like the conventional chemical products, which we'll talk about next. These are, I believe, all the ones that Alexandra tested. The ones in bold are the ones we actually have in Canada. And I've put in the Canadian product names as well. And you'll notice, I think the big thing to notice here is that we have very few products for adults. It's basically just success. Something else that's also a group 5, which is only labeled for landscape ornamentals and not in the greenhouse, which would be closer and delegate together. And pylon, and then TriStar as an option, but I'm always concerned about doing neonics with thrips in case it causes hormesis. And I think Lance may have thought he maybe saw that in the greenhouse. So again, this is why doing whole plant tests over multi-generations is important, because it would be nice to know if we should be using TriStar. Right now, I'm a little hesitant about it. And as Judy's pointed out before, your client or end customer may not accept a neonic anyway, so it's often off the table for us in Canada anyway. So it's really like success and pylon, and then we've got a lot more larvicides. Yeah, so we need to mix in, make sure we're doing a rotation program with larvicides and saving, you know, success and pylon for cleanup at the end and making sure they work by using them responsibly in rotation programs.
We're kind of hoping that the US kind of uses them early and then saves success and pylon for us when we first get them, if anybody's listening. But anyways, so the third line of defense are going to be chemicals. Like, you already have larvae present, you have parvispinus, and you're not seeing any adults, but there's damage there. So you want to probably go into a soft chemistry first. That is a larvicide and going to attack the larva. So again, ferrins, bee-leaf, ryman, contos, and ricar. Contos, you have to be careful. There's probably going to be some phyto on different tropicals, so you need to do testing. But again, you can go into a bio program fairly quickly after these chemistries are used, and they are larvicides that have some kind of long-term effect inside the plant. The fourth line of defense, let's say you get a shipment in and it's already got adults on there, or after you've had them in propagation, all of a sudden adults emerge. You might have to go into a harder knockdown spray, and then you'll have to go into a softer one before you can go back into bios again. But the fun thing about tropical plants is once you start spraying for thrips, it agitates the spider mites. So check your labels, because if you have a chemical that does both thrips and spider mites, you're going to want to use the higher level of the two, because if you're spraying at a spider mite rate which is lower than the thrips, you're going to build resistance for your thrips, and when you need it, it's not going to work. So again, just check that and choose the higher rate for the thrip control. And then some of these chemistries have a maximum number of times you can use per crop cycle. So you almost need to start mapping out what you're going to be doing for sprays, so that you know that if you're going to use pylon, that you don't want to use it at least for 30 days before you use it again. And one thing to remember, if you start using these nasties, you will be finishing with chemicals. So you'll be doing a lot more spraying. Again, you start off with the hard stuff, you got to go wean yourself off with the easier chemistries before you can go back into bios. So finishing, again, we're not worried about larvae at this point. We're worried about the adults. That's what customers see. You might want to use an adulticide plus a larvicide to make it last longer. And an example is Success and Avid. Success does both stages and Avid does the larva. But again, if you've already used these, they might not work for cleanup. And remember, tank mixing is off label. Always test before you spray. Don't do a blanket spray in your greenhouse on all these different tropicals. Tank mixing is always risky, and what works at your facility might cause phyto at somebody else's. It's just the way it is, because every single greenhouse structure is different. So light levels and everything affect your pesticides. So again, pesticides, the important consideration, your stage is important. Don't use an adulticide when it's all larvae that's present. Rotation is key. Again, tank mixes might be more effective, but again, if you only have 4 or 5 chemistries to choose from, and you start tank mixing 2, you really only have 2 that you can choose from then. And you won't be able to do it as a calendar spray program. You can't. So you can't say I'm going to spray every Wednesday, because Wednesday's spray day. And again, spraying threshold is based on your species and variety. And it will, if you only spot spray certain varieties, then it'll help reduce refugia. And again, farm-wide spraying equals resistance.
Yeah, so this is just sort of a depiction of what Judy's been saying. This was, again, work done at that Mandevilla farm when we were doing a chemical program. I wish we were doing a mechanical program, or we started with biological and mechanical control at the beginning. So we didn't spray for a long time, and then we went in with those chemicals in December. And you can see how we rotated. We did Success and Belief at the beginning, and we were able to get 11 to 16 weeks of control, depending on the variety, because they hadn't really seen those chemicals before, or not for a long time. So the whites are in the gray line, and you can see they just continue to be 0. So we just didn't spray them with anything. And instead we focused on the red variety when numbers started to come back up in March, when the temperatures came up. So we switched to, again, something softer, just like Judy was saying, we needed to come out of that period of harsh chemicals with softer chemicals if we wanted to try biocontrol again. So with those softer chemicals, we were getting suppression. We started using biocontrol, numbers were going down, but at that point it was just too close to sale and the grower was a bit nervous. So we cleaned up with Pylon, and that was a successful program. And just to talk a little bit more about that refugia thing, if you've never heard about this before, it's like a tried and true measure of helping to manage resistance in field crops. Usually like in corn, they'll leave like one strip of the corn unsprayed to make sure there's a susceptible population left there, because even just 25% susceptible thrips mated in with 75 resistant thrips will drop your pesticide resistance so much. So if you look at this bottom line in this table where you've got 100% resistance thrips, and they're using like bonkers numbers in these lab assays where they have like 40,000 thrips, and then 2 months later they spray them with the same chemical, nothing happens. 4 months later, barely anything happens. 6 months later, it goes back up. But if you even add in, do the 75 resistance plus the 25% susceptible population, you can see that those numbers are exponentially lower. And then 25% resistant plus 75% susceptible population are even lower. So that's just, this is in the lab, of course, these numbers are a bit, like I said, bonkers, but it goes to show you just leaving some of those varieties where the thrips numbers are low, but maybe not zero, can work in your favor. And also think about how long it takes to come out of resistance. So if you leave some areas of susceptible thrips, you can overcome resistance in just 2 months. If you just keep blasting stuff, you won't be able to spray that chemical again for 8 months. So that might even go into your next years of production if you have any parvi lingering around your greenhouse. So overall, we've kind of come up with an A team and a B team, much like Michigan State does with their pesticides. And really all we're trying to say is, like, if you need to clean something up or you've got a really bad problem, you might want to reach for the A team, but you're going to have to rotate with the B team. The B team has a lot more larvicides in it, some things that are a bit more for suppression. But if you're constantly rotating them with each other or using your monitoring to pick, one that will work for you at that time, you can use all of these chemicals in an effective rotation. And always just, if you're going to tank mix, just make sure that's allowed in your state or province. It's not really allowed in Canada now unless it's explicit on the label.
Mm-hmm. So it's important to know, after we say all this, that just because you spray at the end does not mean the program failed. The goal is to bring the thrip numbers down to tolerable levels and to give you a nicely developed plant so that you can sell it and ship it across the border with no issues. And so it brings us to the final step, and I know everyone knows about this, but not everybody does it. But with these new tropical thrips coming in, the best thing to do is to sanitize your greenhouse. So you want to scrub it with the appropriate cleaner. You want to rinse the cleaner off and then apply a broad spectrum disinfectant at the recommended rate. And then when your greenhouse is clean, don't leave contaminated compost bins sitting in there. Quite often people go for breaks or lunch and then they leave what they're doing and they've got it parked in the middle of a greenhouse that doesn't even have that pest in it. So please make sure that you tidy up. So we're going to summarize everything that we just talked about for you.
We'll be real quick, and then
we'll answer questions.
Yeah, we want to answer questions. But again, if we think of this like a Bemisia program, you want to dip your cuttings at receipt, then reduce your thrips and propagation either with biocontrol or soft pesticides and microbials. Dip your rooted cuttings before potting with oils at a lower rate again, but higher than the initial dip cutting. They can obviously sometimes tolerate a higher level once they've got roots. Initiate biocontrol and mass trapping right at potting. Don't take any breaks, and both of those have to be there the whole time. And delay your pesticides as much as needed, and spot spray varieties if you can get away with it. And you might need to do the pesticides over the winter and then reintroduce bios in the spring if you can. And I think we'll just move on to the bios.
So bios. So this always leads, like, what bios do we use? Now we've talked a lot about everything else, but again, so during all of this trials and figuring things out for us, and in the Netherlands, we found that Aureus is the backbone of these bio programs because, again, it's not specific as to which thrip it feeds on. It will go after a lot of these tropical thrips. Again, jury's still out on the mandevilla crops that come from overseas, again, because of the canopy. Microbials. Bioceres EC is proven effective, but other EC formulas haven't been tested here yet. Again, MET52 is showing some promise, but we haven't done any lab tests. So then the question is, what about other bios? So other big bios, there's a lot of them out there, but as soon as you say the word big bios, you get into big expenses. So the goal is to try and come down to a cheaper one that people can afford. But if we move into some of these other contenders, the price is going to go up. Soil dwelling bios, hypoapsis, aphidia, nematodes, they could be helping, but again, we haven't sort of quantified that or put a number on it. We know it helps for Western flower thrip, and it's great at helping out, but how much is it helping with parvi? And in the mites, we know that Cucurbers and Zwerski are not working in tropicals. Not sure really why, whether it's plant family, poisonous sap, or that real heavy cuticle sap, or heavy waxy cuticle on them. We are trialing degenerins with pollen right now, but again, it's a bigger bio, so it's a more expense. So are these just not worth the cost? And if you take that money and put it into one that works, will that be better? So we're still struggling to find a cost-effective program for each of these crops. What we do know is we need your help and feedback in order to create a program that works here in Ontario. By collecting all the data from everywhere, we can run it in trials and make sure that we have something more efficient to tell you. We need to make sure the chemical rates are followed so that if they work, we know exactly what is applied. And if we're using them at label rates, it will reduce the chance of resistance. And again, we need time and patience to complete these because these plants are very slow growing. So tropicals usually stick around for a while. So it's not as fast as annuals. And we'll need to make sure that we will make sure that the final product you ship will be sellable. So there will be, like Sarah said, that hard cutoff date. If it's not working, we'll intervene.
And lastly, just if you're not signed up to the onfloriculture.com blog already, please do, because that's where I sort of put all my new stuff that I learn about parvi on there. And with that, I'm going to stop sharing, and we've got about 15 minutes for questions before Kara kicks us off and runs away.
Wow, that was great. I can see why you called this webinar Pain in the Parvi. And it's pretty impressive, I think, what you guys have figured out, but obviously still more to learn. So before I get into questions, I just want to mention to everybody, in case you didn't notice, that I dropped a bunch of links in the chat, including the Thrips ID, which I can attest to works. I think Sarah got tired of me sending her pictures of Thrips saying, is this it? So now I've learned my lesson. I can use the key too. Anyways, and there's also a nice PDF. Hopefully everyone can download it from Judy that shows Thrips cycles and everything. So
yeah, and I know we have a lot of information in the session. So just an FYI that this is being recorded and it will be up on the On Floriculture YouTube channel next week once I edit out anything egregious.
Good call. And on that note, actually, one of the first questions is, will you be sharing this as a PDF as well?
I could definitely do that. So yeah, I will put the PDF up on the On Floriculture blog. So if you subscribe there, if you just type in the word parvispinus in the search box, you'll get everything that I've ever done on parvispinus.
Perfect. Okay, so let's jump into questions because there's quite a few. So what about damages in poinsettia? Does it have economic impact in the USA? In Spain, it's becoming a true nightmare.
Great. Oh, thanks. No,
Not yet.
We haven't seen it here yet, but I don't know if I've been any places where they've done crossover of like tropicals and poinsettia, which is, I guess, like a tropical plant with latexy, whatever. So that makes sense. So again, I think this comes down to like workflow and cleanup. Like our growers where they've got hibiscus and mandevilla. Hibiscus are way less susceptible than the mandevilla, but if you put them right next door, even in different compartments, thrips don't respect our plastic barriers and doors and stuff. So when we put them right next door, they get really, really damaged. If you put the hibiscus 2 compartments away and you have like an empty hallway between, the hibiscus are actually quite easy to manage the parvi on with just biocontrol and then like 1 cleanup spray. So I'm hoping poinsettia might be the same thing. And now that we know that that could be a concern, if you grow tropicals and you grow poinsettia, you're going to have to plan out your workflow and your workspace for the next year.
Yeah. Clean up.
Thank you to that person. Send me an
thank
email,
you.
email us.
Yeah.
Send us
Show us
pictures.
pictures, yes, please.
Good to have a heads up. Okay, this question I think may have been posted before you answered it, because I think you sort of addressed this, but for sticky traps, is yellow better than blue? So I don't know if you want
Test it in your facility at different times of year. So there doesn't, so summer versus winter, and even like glass versus plastic. So like in a farm that has multiple locations and they might have different covering types, it might be different for each one of their farm locations. So just test them.
Yeah. We found in the winter that the dark blue are working in the yellow, in the summertime, yellow's working, but both of the greenhouses where we did these trials where we found that are the same structure.
Yeah. And then I found a really dark blue worked at one point and you were finding like a lavender worked. And I was like, oh no, not the lavender. So yeah.
Yeah, it's crazy.
On farm trials.
Yeah.
Okay, next question. Do you think this thrips can reproduce through parthenogenesis, since we find a huge number of female adults in mandevilla flowers?
The key is the washout, because that's where all the males will be coming out as well, right? They're so tiny, you can't see them.
Yeah, I can't remember off the top of my head. I do think it's slightly different reproduction than Western flower thrips, which is Philotocus. And I had looked up parvi at one point, it's fallen out of my head, but it's not, I don't think it's like they're just having females over and over. I think they have to have some males for sexual reproduction in there. Because weirdly, when we put them in a cage, they seem to die out. So it does seem like they need some movement going back and forth. So if it was like an aphid where they're having little clonal females and they can just do that forever, you would have expected the ones that we put in our control cages to be like the most infested. And honestly, they were the least infested. So I think they might actually need to move around and breed. I don't know. So there's a lot of like basic biology work we don't know. So sorry, I can't really answer that one.
Interesting question though.
Yeah.
Okay, so the trap plants, if you spray the trap plants, will you run into resistance issues?
No, probably, because again, the refugia thing, right? So if you're just spraying the trap plant, yeah, you're spraying the most concentrated population, but there's still more out in your crop.
They're going to pull more in.
Yeah, my worry would be if you sprayed your trap plant every week with success over and over and over. So you would want to like rotate it up on the trap plant as well and make sure it's working, because the other issue is the alyssum, like little tiny flowers, little tiny stems. I find it difficult to cover with a contact insecticide.
How do you get in the middle of that, right?
Yeah, and like systemics don't always translocate to petal tissue, so they're a hard one. So maybe just cutting them back, bagging it up, and flipping them out is a good answer. So you got to play with that and just monitor them, I guess.
Yeah.
Okay. In our experience, Western thrips populations drop quickly when the plants are brought outdoors. Is this the same for parvispinus?
I think it will be here.
We did some unofficial testing of bringing some mandevilla and schefflera home. These plants look great in my house, and they were on my back porch. So yes, absolutely. If I could advocate everybody putting their tropicals outside for the summer and then bringing them back in, I think that would be a win.
We know echinothrips doesn't do well outside here because it doesn't have the humidity it needs. So yeah.
I mean, I don't know how the mandevilla would do. Like I can't tell you the growing stuff.
But you know, and if you upset thrips, these are ones that you can upset really easy by washing. So yeah. Does rain do that for us out here? And our temperatures are crazy, right? So, yeah.
Great. Okay. Can you please explain how to conduct a wash for insect collection in greater detail?
Well, I do have.
You need a whole other webinar for this.
Yes,
Yeah.
yeah, we need a whole other webinar. I am making a video. I have all the footage. I just haven't put it together yet. So Caitlin from
Stay
Vineland
tuned.
Research and Innovation Center, if you're watching, that's going to be our little project for the next month.
Nice.
I just collect baggies and I take them home and then I put alcohol in the bag, slosh it around, let it sit for a couple minutes. And then I just, strain it and put it through a coffee filter and
Yeah.
look at it that way under the microscope. So
That's a
that's
good
the
idea.
easy way. But
There's really complicated ways of doing it, and then there's
I
probably
picked a middle of the
some
road,
tricks.
like a little bit more formal than Judy's baggie full of thrips.
Yeah.
And at the coffee filter, I like it.
What kind of alcohol are you using?
Yeah. Drugstore stuff. One for me, one for the
Nice. Okay, next question. Have you experienced any phytotoxicity with using CEF oil at 1 milliliter per liter on any tropicals?
Yes, on Calthea, you have to be very careful. You want to apply CEF oil at the beginning of the day where it has 4 hours to dry, or else you could get phyto if it's not dry in 4. And I never really go higher than 1 milliliter per liter because then you can run into trouble. And then the leaves at Calthea, because they're so flat, it's very hard to get that dry in between. So Calthea's definitely burnt. And again, it's a variety type. And certain mandevilla colors can burn as well. The ones that are slower growing and have a more papery leaf, they have a tendency they could burn. So again, just do a trial first, different times a year, and in different rates with your oils.
What we were doing in one facility was one of the varieties that was very bad. We didn't think we were going to be able to sell anyway. So we started using it as our phyto test kit because the plants were going to be garbage anyway. So, and we actually learned a lot from that and actually rescued the plants and then they were fine.
Yeah.
So
yeah. You'll know if you do an early spray and you get phyto that maybe don't do it later on. They'll grow out of it, but yeah.
All right. Do you have a list of more or less susceptible anthurium species? Or varieties?
So there's a lot of varieties out there that we haven't even gotten in Ontario yet. So just from my experience, it's Ariza in Ontario and there's a lavender one, I forget the variety. But yeah, Ariza definitely, once we got rid of that, it's been a big thing. And then Misto with the Caltheas. So, but that doesn't say it's going to move to somewhere else now.
Yeah.
That we got rid of that one. There might be a new fave. So, but yeah.
Right. Okay. Why do lacewing larvae not work to control larvae?
Well, with the lacewing larva, I mean, it is working, but the trouble is when you start to space your crop out, and then the rates of the lacewings that you have to use is so high. I know lacewings are cheap, but you're starting to put larvae into the crop at very high rates, and then you're going to come along and you do all these sprays, you're going to drown them. I was never able to get good control with lacewing larvae. I mean, I love it. It's probably my #1 bio that I use for everything. But in this case, in tropicals, I could never find them on the leaf. I know they're nocturnal, but could never really find them in the crop, and I couldn't prove they were really doing anything for me. The damage was still there. And I think because they're too big and the parvae are so small, they cannot get down in there early enough before the damage is done.
I have only seen them in the mandevilla flowers when there's lots of adult parvae in there. And so they can just like sit there and have a buffet, but same problem. I can't recover them on the leaves either.
Yeah. Now that's Ontario, like they could be working everywhere else, like that's fine, but this is what we're doing this seminar for.
Okay, in your opinion, how much is the bioseries results based on actual infection by the fungi and how much by its oil base?
We don't know that yet until we actually compare other EC formulations or do a test where you actually like kill the spores but keep the same carrier, that's the only way we would know. So we need some more formalized lab trials for that. So right now we're just going with, that great data that Alexandra put out and going based on that, but yeah, I don't have the mechanism answer for that yet.
And we know there's different oils, like the Laugard M52 has got a different oil, Suff oil's a different one, and so is Bioceres. So again, like we can get burned with some Bioceres, we can get burned with Suff oil, and we can get burned with M52. So, you know, which one is actually doing the work?
Mhm. Okay, would the introduction of younger stages of aureus be more economical and also be effective against parvispinus?
No, can you bring in a younger stage, it's going to diapause right away. It's not going to do anything, right? You need to bring it in when the diapause is broke. In the summer, totally different. But in the wintertime in Ontario, that you're going to bring in a nymph that's really young, it's going to diapause. It has to have been reared past the 4th and 5th end star stage to be effective in your greenhouse.
Okay.
I have
No comment. Couple more questions here. How many generations does it take for parvispinus to become resistant, or are they naturally tolerant in some level?
I don't think we know that yet. Just anecdotally, a grower and I were chatting the other day, and he was saying they seem to develop resistance faster than Western flower thrips, but come out of it faster too. So I would say we have seen resistance to both Success and XRL variants in Ontario, and I saw it with Success after 4 applications. But then again, we were doing a drench, which was, yeah, at the label rate, but when you drench it, it's still a higher volume of the chemical. So if you want to hit resistance, how we do it in the lab, if you want to make a resistant insect, is you blast them at super high rates all the time with the same chemical. So that's kind of what we did. So we sort of like did it to ourselves that first year. So I would be very wary about doing more than 4 applications of the same chemical back to back. And then I might assume you can't use that chemical again for a minimum of 2 months.
And before Dr. Alexandra's charts, all we really had for these tropical thrips was Success. So we were using it. So Success and Ference, you know, you use 2 or 3 applications in a row, and then all of a sudden it's like, uh-oh. So
yeah, the common strategy with avoiding resistance is you never use it more than twice without taking a break of 2 generations of the insect, and then switch it up to something else. You know what I mean? So that's usually the golden rule.
Okay. Aureus is known to be susceptible to Botanigard WP and EC. Is Bioceres EC safe on Aureus?
I don't think it is. I don't think Met 52 is. I think it's susceptible to all those things. Again, these are new strains that are coming out, so you can't necessarily generalize across every species and strain of entomopathogenic fungi, but Aureus is kind of one of those canaries in the coal mine when it comes to microbial pesticides. So I would assume they're not compatible, but you can use them at different stages. And especially if, you know, you're drenching Met 52, but your Aureus are on the leaves, you could time those introductions.
Yeah. So, and it's no matter what, their contact. So if an Aureus is sitting there and you're going to spray with any oils, you're going to kill them. And their nymphs are really susceptible to the Botanigard or the active ingredients on them. So adults, not so much, but yeah, it's contact. So again, don't put your bios there and then spray the next day. Like it's all, and again, it's all, we're not blanket spraying here. We're only spraying as needed on certain varieties. So that allows other populations that you have in the greenhouse to continue.
And if you're doing weekly applications of Aureus, right, you can do your weekly applications of Botanigard or Bioceres on a Monday, and then be releasing your Aureus the next week and stagger them that way. You know, there's ways around it.
Yeah.
Minimize your losses there.
Yep.
Okay, has Contos been trialed on Anthurium?
Yes, it has, I believe. I can't tell you the rate. I'd have to look that up. But again, you know, I had the customer pull one from every single generation. Now, with Contos, 1 application could be okay, but 2 would be where the phyto happens with Contos, because it does stick in the plant a long time. So this is where you're going to really need to watch your Contos applications that go back to back.
We got 2 more questions to go.
Okay.
Is there anything new regarding lures to make pesticides more effective?
So that's why that was at the beginning of my slide.
Those
Can we
lures.
make these scent globules? Can we take them from greenhouse banded, and then, because they're huge, can we make a big pheromone pot of that stuff that will make parvispinus disappear? Like, we need to do trials like this. Or
Yeah, I've done some really preliminary work with the lures that smell like plant defensive chemicals, because the other lures are Western flower thrips sex pheromones. So those ones won't work. But the ones that smell like plant defenses could work. And granted, I tried in winter when it's harder for like volatiles to move through the greenhouse, and I didn't really see anything, but wouldn't rule it out. I think, again, we just don't know yet. And it is really hard to do experiments with volatiles in an open working farm system.
And we, 4 or 5 years ago, we never had this many Thrips species we had to worry about, right? So yeah, some of them don't play nice with each other. So if we can upset the other guys and not have them come in, then I'm all for it.
Would be interesting. Okay, the last one is actually not a question. It just says, thank you, great presentation, very informative and useful. So
that's a
you.
great
Yeah,
note to end on. So
what a way to,
Yeah.
way to end.
well, thanks everyone for listening to both of us try to cram 3 years of research into that.
Yeah.
And yeah, check out the blog and we'll keep posting stuff there, and maybe Judy and I will write a magazine article or something
Yeah.
soon.
And if you have any more questions, the research is ongoing. We're going to get together at the end of this next Mandeville Anthurium crop and put more data together. So yeah,
Great.
we'll figure it out.
Okay, thanks
Thank
everyone.
you. Bye.
Bye, everybody.